Welcome to Lecture 4, the final video on enzymes. In the first three videos, we covered everything: what enzymes are, how they work, how temperature and pH affect them, and how to carry out enzyme experiments. Now we put it all together. This video is a complete past paper walkthrough. I will show you real exam style questions, the exact types, Cambridge sets, and solve each one using mark scheme language. By the end of this video, you will know exactly how to answer every type of enzyme question. So let's go. Let's start with a multiple choice question, paper 1 style. Read the question. What explains why a reaction controlled by an enzyme happens more slowly as temperature decreases? The four options are given. Let's eliminate wrong answers one by one. This is the key MCQ strategy. Now look at option B. Option B is wrong. The enzyme is denatured. Wrong. Denaturation only happens at high temperature, not low. So eliminate B option immediately. Now let's look at option C. The enzyme is killed by the low temperature. Wrong. Enzymes cannot be killed. They are proteins, not living things. Classic examiner trap. Eliminate C. Option D. The substrate changes shape and cannot fit into the active site. Wrong. At low temperature, neither the substrate nor the enzyme changes shape. Shape only changes during denaturation, which requires high temperature. Eliminate D option. Now the correct option is option A. At low temperature, molecules have less kinetic energy. They move slowly. Substrate collides with the active site less frequently. Fewer enzyme substrate complexes form per second and rate of reaction falls. Answer is A. Every time you see killed or denatured at low temperature in MCQ options, eliminate them immediately. Now look at the question type 2, structured question, paper 2 style. Amylase is an enzyme. A student added amylase to a starch solution at 60 degrees centigrade. After 10 minutes, starch was still present. Explain why. 3 marks. Command word. Explain. Give biological reasons why. So the model answer is 3 points, 3 marks. Mark 1. 60 degrees centigrade is above the optimum temperature for amylase. Simple first point. Always state this. Now mark 2. The high temperature caused the active site to change shape. It is no longer complementary to the substrate starch. Two keywords here are active site and complementary, both worth marks. Mark 3. The enzyme is denatured. Starch cannot bind to the active site. No enzyme substrate complex forms. Notice this answer has 5 syllabus keywords: optimum, active site, complementary, denatured, enzyme substrate complex. Everyone is in the mark scheme. Common student mistake is writing the enzyme was killed by the heat. That gets 0 marks. Use correct biological language. Now the question type 3 is graph question. Very common in paper 2. The graph shows the effect of temperature on enzyme activity. Describe and explain the shape of the curve. 4 marks. Two command words are here: describe and explain. You need both what you see and why. Model answer is 4 marks, 4 points. Mark 1. Describe the rising section. That rate increases as temperature increases from 0 degrees centigrade to 37 degrees centigrade. State actual values from the graph. This is describe. Mark 2. Explain the rising section. Because kinetic energy increases, more frequent collisions between substrate and active site. This is explain the biological reason. The Mark 3 is describe the peak. Rate is highest at the optimum temperature around 37 degrees centigrade. Mark 4. Describe and explain fall. That above 37 degrees centigrade, the rate falls sharply. The enzyme gets denatured. The active site changes shape. The substrate cannot bind to it. Remember, describe means say what you see. And explain means give the biological reason why. Both together give you full marks. Now the question type 4. Data table question. A student tested amylase at different pH values. Use the results to describe the effect of pH on amylase activity. It contains 3 marks. Look at the table carefully before you answer. Find the pattern. At pH 4 and pH 9, starch did not disappear at all. This means amylase had no activity. It is too far from the optimum pH value, so the enzyme gets denatured. As pH increased from 5 to 7, the time decreased from 20 minutes to just 4 minutes. It shows shorter time means faster reaction and higher enzyme activity. At pH 7, starch disappeared in only 4 minutes. So this is the fastest reaction. This is the optimum pH for amylase. Above pH 7, like at pH 8 and 9, time increased again because activity fell down. So model answer is at pH 4 and pH 9, starch did not disappear because enzyme had no activity. Mark 2. As pH increased from 5 to 7, rate increased. Shortest time at pH 7. So pH 7 is the optimum pH value. Mark 3. Optimum pH is 7, highest rate of reaction. Above pH 7, the rate gets decreased. Key tip. Always quote values from the table. Do not say rate increased. Say rate increased between pH 5 and pH 7. Specific values equal more marks. Before we finish, 4 golden rules for every enzyme question. Rule 1. Never write killed or died. Always write denatured. Say the active site changes shape permanently. This distinction alone can earn you 2 marks. Rule 2. Always use syllabus keywords: active site, complementary shape, enzyme substrate complex, denatured, optimum, kinetic energy. If even one word is missing when it should be there, you lose that mark. Rule number 3 is quote values from graphs and tables. Never say rate increased. Say rate increased from pH 5 to pH 7. Specific values equal marks. Rule 4. Read the command word first. Describe means say what you see. Explain means say why it happens. If the question says describe and explain, do both. Students lose marks constantly by explaining when asked to describe. So, 4 rules, every enzyme question, guaranteed marks. And that is chapter 5, all about enzymes, completely done. Let's look at what you covered across all 4 videos. Video 1, what enzymes are? Biological catalyst, active site. We studied about enzyme substrate complex and specificity of enzymes. In video 2, we covered temperature and pH effects. Optimum, kinetic energy, denaturation, graph reading. This is all what we studied. Video 3, we covered practical skills, variables, experiments, buffer solution, repeat and mean. Video 4, past paper skills, MCQ strategy, command words, quoting values, keywords, all were covered. You now have everything Cambridge can ask about enzymes. Every question, every enzyme technique, every mark scheme keyword. The next chapter is coming very soon. Subscribe so you do not miss it. And share this video with any IGCSE Biology student who needs help with enzymes. See you in the next one.